Assalamu alaikum and good morning, students. I hope all of you are doing well. After a short break, it is biology for class 10, and today I am going to start practical. First, before explaining the practical, let me uh, recall you what is the syllabus for the biology this year. There are the 4 chapters: life processes, how do organisms reproduce, from heredity and evolution only heredity is there, and our environment. The chapter management of natural resources is going to come in internal assessment, and the chapter control and coordination is deleted for the assessment, but I have already explained you. So that chapter notes point I am not going to give. Okay, remaining all chapters I have completed, the notes, exercise, everything I have completed for you. Now the practicals, there are only two practicals for biology. One is to show the carbon dioxide is given out during the respiration, and the second one is study of permanent slides of binary fission in amoeba and budding in yeast. Okay, students, so this is the syllabus for the class 10 this year. Now today's practical, that is to show that carbon dioxide is given out during the respiration. And students, you have to write this practical in your practical notebook. Okay, similar way, first you have to write the aim, then you have to write the materials required. So what is the aim? To show that the carbon dioxide is given out during the respiration. The materials required are one conical flask, U-shaped delivery tube, cotton wool, thread, beaker, test tube, water, rubber cork, and freshly prepared KOS solution. KOS means potassium hydroxide solution, Vaseline, and germinated seeds. Okay, so these are the materials required for to perform this practical. Now, this is the pictures. What are the materials? This is called conical flask. These are germinated gram seeds. This is U tube. You can see it is banded at the rectangular at the two sides. Then this is a small test tube. It is the thread. This is cork. This is beaker with the water. And in this small tube there is freshly prepared KOH solution. So these are the material required. Now procedure. What to do? First you have to take about 25 to 30 seeds of gram. Gram means chana, and germinate them, this seed, by the placing them on the moist cotton wool or by moist blotting paper. So we can take anything. Either you can take the cotton wool or blotting paper. But what is necessary? Moist. Moist means the water should be there. Clear? And we will observe after 3-4 days the germination will start. Now place the germinated seeds into a conical flask and sprinkle a little water in flask to moist the seeds. Okay. Number 3. Take freshly prepared 20% KOS solution in a test tube and hang it in a conical flask with the help of a thread. Close the mouth of conical flask by placing a rubber cork containing one hole. Okay, so these are the steps. Already I have shown you the figure. This is the figure. First you have to take the gram seed in the conical flask. The gram seed should be germinated. Then you have to put the chaos solution in this small test tube and tie with the thread, and you can means with the rubber cork you can hang it over here in this way. Now through the hole of rubber cork, insert one end of the U-shaped glass delivery tube in the conical flask and another end in the water. And then we have to seal all the connection with the Vaseline. Why with the Vaseline? Because we have to prove that carbon dioxide is given out during the germination or during the respiration. Okay, and so that the from the any hole it should not come inside the conical flask. So we have to make this apparatus airtight. Now we have to mark the initial level of water in the U tube here. We have to mark, okay, this is the level of the water. Then next point, keep the apparatus undisturbed for 1 to 2 hours and note the change in the level of water in the delivery tube. So student, I think procedure you understood. What is the procedure step? First you have to take the gram seed that should be germinated in the conical flask. Then you have to take a small tube containing the potassium hydroxide. You have to tie it with the string. Then you have to take a rubber cork. From rubber cork you have to pass a glass tube, one end in the conical flask, another end in the water, and water is in the beaker. Okay, and then you have to make this apparatus, this setup airtight by using the Vaseline. And then we have to mark the initial level of water in the U tube. Now what is the observation? After 1 to hour, we will see, you can see over here, the level of water is increasing. Why it happens so? Okay, so what is the observation made by us? That after some time the level of water in the U-shaped delivery tube in the beaker is rises. What is the result? Why it happens so? Because the germinated gram seed in the flask release carbon dioxide. It is carbon dioxide 2 is missing during respiration, and the carbon dioxide release is absorbed by KOH. Here the KOH is captive because KOH is potassium hydroxide is absorbing the carbon dioxide. Okay, so what the carbon dioxide is released during the respiration, it is absorbed by carbon dioxide. So that space is empty over there in the flask, and it creates a vacuum. This creates a vacuum in the flask, and it causes the movement of water in the delivery tube. What happen? If there is a space, there is a space in the conical flask, so that vacuum is creating the water level to rise in the U tube. Understood? And that's why the water is moving inside the, or the level of water is increasing in the delivery tube. So this is the experiment completed. Now the precautions are there. What the precautions we have to keep? The first precaution is that the germinated seed what we are taking should be kept moist. They should be kept moist. This you are studying from the beginning only, that the for the germination what is required? Air and water. Okay, so air they are getting from atmosphere and they should be moist, then only the seed will germinate. The second one is all the connection of the setup should be airtight, then only we can give this conclusion that the carbon dioxide is released during the respiration. Okay. The next one is the chaos solution should be used. It should be freshly prepared. It is not that long back you are preparing the potassium hydroxide solution and then you are using for this experiment. Then you will not get the satisfied result. The next is keep one end of the U-shaped delivery tube in conical flask and the other end immersed in the water of the beaker. You have to ensure that the U-shaped tube should be the other end should be in the immersed in the water. वो it should be immersed, यानी वो पानी में डूबा हुआ होना चाहिए. What would happen if it is not inside the water? Then obviously the air will enter through that tube and you are not able to perform the practical because the air will enter through the tube. And second, then how can you find out the water level is rising? Understood? And then the last point is there, that is the test tube containing potassium hydroxide should be hung carefully. Okay, it should not happen that the potassium hydroxide is falling down in the conical flask, or by chance in between the experiment it fell down, it should not happen. So it should be hung carefully in the conical flask. So student, this is the complete experiment, experiment number 1 of biology, and that experiment aim was carbon dioxide is released during the respiration. As I already told you that you have to write this experiment in your practical journal. So, and the diagram is also compulsory. One diagram only you have to draw over there. That is the complete setup. Steps you have to write. That is the aim, materials required, then the procedures, all point you have to write. Then you have to write the observation. Then you have to write the result, and then it was the precautions and along with the diagram. So write all these things in your practical notebook. And then the last practical is that is the study of the permanent slides of reproduction in amoeba and in the yeast, that I will explain in my next video. Till then, have a nice day ahead. Take care of yourself and remain connected to the learning process.